Hi, and thank you for tuning in to the Charles E. Holman Morgellons Disease Foundation's YouTube channel. I'm your host, Jeremy Murphy, and today we're very excited to have microbiologist Marianne Middelveen joining us for a conversation. Marianne Middelveen is a microbiologist and medical mycologist in Calgary, Alberta, Canada, currently working in the field of veterinary microbiology and specializing in bovine mastitis. She received postgraduate degrees in microbial and biochemical science and in environmental science from Georgia State University and University of Calgary, respectively. She has been involved in research projects at the Centro Amazonicano para Investigación y Control de Enfermedades Tropicales, Porto Ayacucho, Territorio Federal Amazonas. Gosh, did I get that right, Marianne?
Well, we could translate it to English. That might be easier. It's the Amazon Center for Research and Control of Tropical Diseases in
Wow.
Puerto Ayacucho, Venezuela. That's a little bit easier.
Well, that's commendable. I'm glad you understand the language. I totally messed that one up. Should also note that you did work for the Centers for Disease Control in Atlanta, Georgia.
That was part of my thesis research when I was a grad student at Georgia State.
Commendable. And currently, you've been involved in the research related to Lyme disease on behalf of the Canadian Lyme Disease Foundation.
That was quite a while ago. We did a survey that yes, I did some volunteer research work for them and of course my volunteer work on Morgellons and Lyme disease.
So how are you doing?
I'm good. How about yourself?
Well, I'm a little nervous being in this clean room and in the suit and everything, but we'll manage. We're going to make it. Thank you for asking.
Okay.
Marianne, and we are live on YouTube. Everybody, glad to see you over there in the chat. You've been studying Morgellons disease for a long time, and you've produced a lot of research, contributed to a lot of research regarding establishing facts about the disease. Can you tell us now, today in May 2020, what are Morgellons fibers and how are they made?
Well, Morgellons fibers are the key diagnostic feature of the condition that we call Morgellons. And these fibers are unusual microscopic fibers or filaments. They are formed by human cells and in human skin. And they're usually colored red, white, or blue, or sometimes black. And they're formed in the presence of a pathogen or pathogens.
What pathogens typically are involved in Morgellons disease?
Well, typically we see the key etiologic pathogen that we see is our Borrelia species. So we see different species of Borrelia in the skin. But there can be other pathogens. Let's see, Helicobacter pylori has been detected, as has Bartonella henselae been detected in skin, and also Treponema denticola and a rickettsial species.
So if Morgellons presents, then there's going to be some infectious agents involved.
We feel that we have strong evidence linking Morgellons disease to an infectious process, and that it is the result of an infectious process.
Commendable. Marianne, what is a follicular cast, and how is that different from a Morgellons fiber?
Okay, a follicular cast is an accumulation of keratin around the hair shaft of a hair follicle. And it's indicative of follicular keratin production. It indicates a defective process in keratin production. And Morgellons fibers are elongated hair-like structures, and they sometimes form around this thickened material, although not always.
So these are two different symptoms, but are they both particular to Morgellons disease?
Well, follicular casts are not specific to Morgellons. They can be formed, result from other conditions. So, although they're frequently found in Morgellons patients, they're not exclusive to Morgellons. Whereas the Morgellons fibers are the key diagnostic feature of Morgellons. That's how we define Morgellons. These unusual filaments that are microscopic and are found in the skin, we feel that's the key diagnostic criterion for Morgellons disease.
Okay, got it. Well, so do different pathogens cause follicular casts and uh, as opposed to Morgellons fibers?
Follicular casts can probably result from many different conditions, including infection. So I'm not an expert on follicular casts, so I wouldn't know exactly, what other conditions it might be associated with. But it's not an exclusive process to Morgellons or to infection with Borrelia.
But the fibers, the microscopic fibers, certainly are.
They certainly are. That's how we define Morgellons disease.
Is it true that one symptom related to Morgellons is that patients' hairs will grow in reverse? That is to say, instead of out and away from the skin, the hair will grow in down towards the muscle. Is this true, and how far down do they grow?
Well, of course, it's not all hairs, but occasionally in some patients, we do see these unusual hairs that grow from thickened follicles, and they're growing from the wrong end of the follicle. So instead of the follicles, usually like a little cup shaped thing, and usually the hair grows up from that and then protrudes through the skin. Well, in some patients, we see these thickened follicular casts, and from the lower end or the bulb end, we see a hair growing downwards into the dermis. So we do see that in some patients, but not all Morgellons patients have that feature. It's just a common finding that we find in Morgellons skin specimens.
Sounds like that would be especially painful.
I think so. From my understanding, the patients say that when they have this condition, they describe it as a sensation that of a needle or a knife-like sensation. And certainly, you know, humans get ingrown hairs and they can become very inflamed, and ingrown hairs can be very, very painful. We also see, besides these hairs, we actually, in some patients, we see just filaments that are not associated with the follicular bulb. And these filaments also can grow down into the dermis. And what's interesting about those particular filaments is that when we stain for Borrelia, do a specific stain looking for Borrelia infection, those fibers that grow downward stain strongly positively for the presence of Borrelia. So, and these are very unusual filaments. So I think it's really interesting that we have shown a direct association with Borrelia infection and unusual fiber formation.
So you have this chemical stain that shows you if a particular bacteria is present. Is that the antibodies for that bacteria, or is the stain looking for the pathogen itself?
We use a technique called immunohistochemical staining, and then when we do that technique, we are using an antibody that's specific for a particular target.
And what you're saying is that the actual structure, the hair itself, uh,
The fiber, yeah.
it
You know,
actually,
hair has a certain structure. So in order to be called a hair, it has to have some scaling, like human hairs have scaling, they have a bulb, they have a cortex, they have a medulla, and some of these fibers lack some of the characteristics. We just see, fibers that are starting at the basal cell layer, which is a deep layer in the epidermis,
right.
and these fibers are formed from skin cells from that layer growing downwards. And uh, these particular structures do have a little bit of a medulla, and they do have a cortex. So the medulla is a little hollow center, and then the cortex is the solid part of the fiber, and they do grow downwards. So in terms of fibers, I would just call them a hair-like fiber.
Gotcha. And these are the Morgellons fibers that we're discussing right now.
They are some of the Morgellons fibers. There's others that are, in some patients, I see little tiny microfibers, and those ones do not have the medulla in them. And those tend to be, those tend to originate from a follicular, like from a follicular bulb, or a bulb that is like a follicular bulb, and they tend to grow upwards. So we see a lot of unusual things in Morgellon skin, and some of which are the unusual filaments, and there's other unusual objects that we do see as well.
And you're saying that the research demonstrates that these fibers themselves stain or react to the Borrelia stain. So,
Some of the unusual fibers do. And at the base of attachment, when I look at them under high magnification, I can see that these are formed of keratinocytes, which is a certain type of skin cell that produces keratin. So I can see that there are Borrelia infected keratinocytes at the base of attachment of these fibers.
does that mean that the fibers themselves are partially made of bacteria?
Well, they contain bacterial substances, but the primary chemical component in them is probably keratin or collagen. But it would be, it will have some Borrelia in them. Yeah.
Oh, okay. So the genetic material is just there from where the bacteria
And
has gone
mostly
inside this.
human skin cells. I mean, that the fibers are mostly composed of human skin cells. And then, I mean, like hair, as hair or these fibers grow, in the older sections, they tend to lose their nuclei, just leaving the keratin or collagen. And then, but in the base of attachment, we can see that a lot of these fibers are composed of keratinocytes. Those are the cells that produce or make keratin.
Right.
And those cells tend to have intracellular infection of Borrelia. So yes, there's a component of, there's some Borrelia substances in these fibers, but I wouldn't go as far as to say the fibers themselves are composed of these of that bacterium.
Yeah, it's just it makes yeah, and thank you for clarifying that. It's not that the fibers themselves are made up of the bacteria; they're just the cells that make the fibers have had infections inside of them.
Right, right.
Okay,
So when we're staining for it, wherever there's these Borrelia proteins present, it'll light up a kind of red color, and we can see that those particular cells are red staining.
okay,
Okay.
that actually does clear up a lot about that. And what we're talking about is real science. I mean, you did the work to have this research published in journals and scrutinized by your peers, right?
We did, and like any good science results from controlled experimentation.
Oh,
So all of our experiments are controlled. We have positive and negative controls. We make sure that our findings and our conclusions are based on solid scientific evidence.
um, well, let's shift gears just a little bit for a second.
Okie dokie.
Is it true that some Morgellons patients do not have any sores at all?
I've heard of patients that don't, and some of these patients do have unusual filament formation without an actual sore or ulceration. But I'm sure if you were to take a biopsy from that site, you would see some unusual things happening inside that area. So there'd be some unusual pathological findings.
Gotcha. So maybe not a sore, but definitely not normal by any means.
No, I mean, maybe you'd see some Borrelia infection in there. Maybe you'd see some infected keratinocytes. There'd be probably something happening in that area.
Um, Treponema denticola is been demonstrated to be associated with Morgellons in some previous studies. Is is that
We
pathogen?
have found Treponema denticola in some specimens, but we've never found it alone without Borrelia infection.
Oh, so you find it some of the time, whereas you find Borrelia all of the time.
Pretty much all the time. It depends on the study and what detection method we use. But pretty much if you use different detection methods, we can find Borrelia in the skin of patients. It's not to say that hypothetically a species of treponema couldn't cause it, because we don't in fact know that that's not true. But from what we see, pretty much all the patients have Borrelia infection. In some of our studies where we've used only one detection technique, we're not 100% successful in finding Borrelia in all of them. But, if you use different primers for PCR, which is a molecular technique, and then you use PCR, plus you use the immunostaining, plus you use serology. And if you use different techniques, you can pretty much find that pretty much all Morgellons patients have Borrelia infection that's detectable by some assay or another.
Okay, and Borrelia is not just Lyme disease; it's also relapsing fever as well.
That's true. There's around there's at least 40 species of Borrelia around.
Oh,
So, and some of which are thought to cause disease in humans, and some of which are not thought to cause disease in humans. But there's a
oh,
great genetic diversity of Borrelia species, and many of these are capable of causing infection. And detecting, a detection technique that works on one species might not be effective for another species. So detecting Borrelia becomes is can be very difficult and complicated, because you can't use just one assay to identify all species.
and is it possible that there are some species of Borrelia that we just don't have the technology to detect at all yet?
Well, it's possible, but most of them are going to be detectable if you use the correct or if you use, different techniques, and it, depends on the specificity and sensitivity of the test. And like some things like immunostaining, I pretty much see if I use an immunostain that has a broad specificity for both treponema and Borrelia, pretty much all the patients come out positive on that assay.
That's fascinating.
I haven't seen any that were negative on using that assay, using an antibody that has a broad specificity.
Okay.
But because that isn't as specific, then we also do other assays to try to genetically identify what kind of Borrelia we have in the sample, to make sure that it's not just contamination from some cross-reacting bacterium.
Yeah, that's what I was getting at with the questions regarding treponemes. Treponema denticola. Well, first, is Treponema denticola any different from Treponema pallidum, the causative agent in syphilis?
It certainly is. I mean, they're both members of the genus Treponema, but one is associated with periodontal disease, and it's pretty common. And the other is, as a Treponema pallidum is associated with syphilis and is the causative agent of syphilis. So, definitely different organisms.
The cross reactivity. Do treponemes cross react with Borrelia?
They can, and again, that depends on what antibody you use. If you use for example, a monoclonal antibody that's targeted to a very specific protein found only on Borrelia, but not on Treponema, then it won't cross-react with Treponema. But in some of my experiments, I use a polyclonal antibody, and I did that initially on purpose because I knew there was a great genetic diversity. So initially I didn't know whether I knew spirochetes were involved, but I didn't know if it was Borrelia or Treponema or both. So initially I used something with a broad specificity that targeted both Borrelia and Treponema. And then, we also use monoclonals that are very specific for Borrelia, and they don't cross-react with Treponema. So it depends on the specificity and sensitivity of your reagent. And when you do controlled experiments, you have to determine what your antibody reacts to and what it doesn't. So, for example, our antibody that reacts to Treponema and Borrelia, it doesn't react to E. coli or other common gram-negative bacteria.
Oh.
So, and it doesn't react to gram-positive bacteria such as staph or streps that are commonly found in skin. So, when we see that, we know, pretty much that it's a spirochete in there.
So you didn't just say, I think this is Lyme and we're going to go with that. You actually conducted experiments and implemented technologies that would show you whether it was treponemes or Borrelia.
Exactly. And we don't just use immunostaining. We also use PCR amplification, which will detect the presence of DNA. And then if we detect DNA, then we can go ahead and have that DNA sequence, and we can tell in many cases what species of Borrelia we are seeing.
Okay. And besides PCR and immunostaining, do you implement any other technologies to distinguish the pathogens?
We've used Dieterle staining, which is pretty non-specific. It will stain other bacteria, but it allows us to see the morphology of spirochetes. We used electron microscopy, and we could see definite spirochetes with electron microscopy. We used a method called fluorescent in situ hybridization, where you use a DNA probe that looks specifically for the DNA of Borrelia. And, again, we ran that with controls, and we learned, that the ones that we used were pretty much specific for Borrelia burgdorferi, one of the Borrelia species.
Wow.
Again, you every time you do an experiment, you want to know and understand exactly what you're seeing, and you want to understand the specificity and sensitivity of your assays and the limitations.
Well, and you're also being peer reviewed, so your peers are scrutinizing your work, and if they see anything that would make your conclusions invalid, then they're going to keep you or prevent that research from being published, right?
Definitely. And some of them, they'll bring up, like there was one reviewer in one paper who was concerned that we were just having non-specific sticking of antibodies. And so because the skin was inflamed, he said, well, inflamed skin might just be sticky. So, how do we know that it's just not the antibody sticking to skin in general? So he requested that, we do some staining with skin that had other conditions. So, I used skin that had psoriasis, and then I used skin that had fungal infections. And then we looked to see if there was reactivity with the antibody, and there was not. So, the peer review is good because it keeps the science honest, and you have to go through a great deal to get a paper peer reviewed and published.
And as a mycologist, you haven't found any evidence that Morgellons is in any way associated with fungal infection, right?
Definitely not. I have not seen any fungal elements in Morgellons skin. It's not to say that a patient couldn't have both conditions. It's just I have not seen it, and I've seen hundreds of Morgellons samples. So, fungal infection does not appear to be a problem or an etiologic agent causing that condition.
Is Lyme disease demonstrated to cause ulcerations like syphilis?
Well, I think Morgellons is a prime example. We have one example there, but Lyme disease is associated with a lot of dermatological conditions. So if you were to just go onto PubMed and do a search on dermatological conditions and Borrelia or Lyme disease, you would find that skin inflammation and skin conditions are not uncommon phenomena in Lyme patients.
Gotcha. What has your research shown you about Morgellons in dogs? Is it different from Morgellons in people?
We have found a condition that was analogous to Morgellons in dogs, and the dogs had the same unusual filament formation, and they also had detectable Borrelia infection. So pretty much it was identical to what we see in humans.
Wow. So dogs, is it Lyme disease in dogs as well?
From what we saw, yes. But I'll say that hypothetically, we don't know if treponema infection could cause a similar or identical condition in humans and dogs. We do know, for example, that Carl Ekbom, who described delusions of parasitosis, in his original case study, those cases he described were very similar to what we call Morgellons. There was a lot of similarity. So it may have, in fact, there may have in fact been some unusual filaments in these patients. We don't know. But certainly there was similarity. And in his study, 3 out of 7 patients had known cases of syphilis. And then there was a French scientist, Vier, who published similar case studies, and 6 out of 8 of his patients had known cases of syphilis. So I'd say hypothetically it's possible, and maybe those early cases were caused in fact by syphilis. We'll never know, but it's interesting to hypothesize that it's possible. And nowadays, syphilis is identified and treated more quickly, and perhaps it hasn't had the chance to evolve into, a Morgellons-like condition in patients.
Now, Ekbom didn't have the benefit of PCR, right?
No, he didn't.
Okay.
He didn't. So, and and he also maybe he described little grains of sand and things in the specimens and some filamentous things, but he didn't have the techniques that we do to show where these, to show what the fibers were composed of, what the origin of the fibers were. And so and these unusual hairs, when they're detached from skin, they do look like lint or dust particles. They even unless I see them deeply embedded in skin, I don't know that they are in fact not contaminating fibers.
But when you do a chemical analysis on it, it becomes apparent that they are biological in nature.
Well, when you can see the actual cells in which they're coming from, and you can see what cell layers they're coming from, yes. And when we use a stain called Gamori trichrome, we can see that in fact these fibers are all composed of keratin and collagen, and which aren't, those are proteins that are found in other birds and animals and organisms, but we know for a fact where the cells in which these unusual fibers are originating from. So we've got a pretty clear idea of what is happening in the skin.
Yeah, I mean, it sounds like based on what we just talked about over the past 30 minutes, that anybody should have a clear understanding that Morgellons fibers originate from inside the body, from uh, cells that produce keratin and collagen that are infected with Borrelia bacteria.
I think if a person was inclined to actually read and objectively understand the research, I think it would be clear to them. So they just have to give it a chance. They have to sit there and objectively read and objectively evaluate the evidence.
Now, the they have to be open to the potential that the fibers are not lint, and that has held back a lot of people from even achieving respectable research. Is that they didn't consider that there could be a biological process which can create these lint-like fibers inside the body. And that's kind of what happened with the CDC study, right? They
Well, they
start.
actually found cotton fibers in there, and that was because they weren't very careful about how they took their samples, and then they didn't have a clear diagnostic criterion that we use the presence of these unusual fibers, and they have to be projecting or underneath unbroken skin so we can be assured of the origin. Whereas they just, if you look at their diagnostic criterion, it was quite different. And they just went through their database looking for people with delusional parasitosis or people who claim to have fibers, and um, and then there were a lot of problems with their study. And then so they chose basically the wrong patients, they took the wrong samples, and they came to the wrong conclusions. And they cleaned, when they took biopsies, for example, they used cotton to clean off the skin, and then they admitted, if you read their paper, they admit that they possibly introduced the cotton fibers themselves. It's actually in there.
And so that's a big difference. Is that your research was controlled in such a way that those contaminants could not have been introduced into the specimens.
I do sometimes see contaminating debris in samples. For example, I see some plant material, but it's important to only study what is actually inside the skin under unbroken layers.
Attached at the basal layer.
And then in Ekbom's study, some of the patients were presenting with collections of material that they had collected, and he had no way of properly analyzing or determining, and nowadays we recognize that a lot of mental conditions result from the infectious process, and in those days it wasn't widely accepted that this could happen, and so with Morgellons we have it historically being associated. What we call Morgellons is historically associated with those early case studies by Ekbom.
Yeah.
And so it's so ingrained that it's difficult to turn doctors around. They've been taught for decades that symptoms like Morgellons are really forms of delusional infestation or delusional parasitosis, that it's hard for them to get their brains around to accept new evidence.
And a person can have both Lyme disease and syphilis at the same time.
They certainly could. Just like, some patients seem to have Lyme disease and treponema Denticola infection,
right.
too. And in those patients, I think probably the infection in the skin is probably originally Borrelia, but people put their hands in their mouth and then they scratch their skin, and probably the same with Helicobacter too. They probably introduce some of these bacteria by scratching.
And as limited as Ekbom, the technologies available was to him at the time, his research was fairly thorough and descriptive. And I think
it was, and he actually believed that there was an underlying cause to this. He didn't just say these patients are, mentally ill. This is all in their head. He felt that there must be an underlying cause. Right. And he said it was important to try to identify the underlying cause. And our doctors today love to jump to Ekbom, but they are forgetting that Ekbom felt the underlying cause was important. A lot of our doctors don't seem to think finding the underlying cause for this is in fact of any importance at all. They're just inclined to, blame it all on mental illness and say, okay, patient is delusional, give them an antipsychotic and be done with it.
Yeah, unfortunately, in many places around the world, it's not readily accepted that infections result in mental illness or behavioral concerns.
Which is ironic when you consider that, syphilis has been associated with mental illness, and that's historically documented. So you'd think that, people would be more open-minded to this possibility, and it's quite distressing that they're not.
Yeah, it's almost like we entered a completely different reality somewhere in the past 100 years, where all that literature we have about how syphilis can affect a person's behavior is just forgotten.
And like Morgellons, and in syphilis, we see that not all the patients with syphilis have mental illness. Right. And the same with Morgellons. There's many, most people with Morgellons are, not, they don't have any kind of delusional mental illness. And a few of them, what they might misinterpret some of their symptoms. Like they have a sensation of movement, which, in medical terms is called formication, and they might mistake that for a parasitic infection, but that's really more of a mistake. Or they see these unusual fibers and they mistakenly believe that that is a worm. That's also a mistake. That is not a delusion. A delusion, is not based in reality.
The plugs, the white worms. This is basically what the wart-like structures that's involved in bovine digital dermatitis. Is this correct? Can you elaborate what?
No, the little plugs that I see, they're more like a little whitehead, or they'd be just like a little bump under the skin. And those are, the follicle, like I said, is a little plug, and inside it can be, keratin and sebum and different things can accumulate. And those kind of plugs tend to be quite common in Morgellons patients, and sometimes they pop out, and then under the microscope they look like a little seed-like thing, and so people call them either they think it's a grain of sand or a seed, or some patients think they're cocoons because of the they tend to be shaped sort of like that, with a little head, like a little pointy, almost teardrop shape. So, some patients mistakenly believe they're a cocoon or something like that. And then if you read Ekbom's paper, it's interesting because he says there's sand-like particles in those samples too.
Yeah,
So I find it all very interesting.
those can often be gelatinous in presentation.
Sometimes a jelly-like substance does come out of the skin, and I've sectioned some of that, dried it and sectioned it, and I found a lot of Borrelia spirochetes inside that gel. But yeah, I suppose some of those little cocoony type things, or the plugs, could be a little bit gelatinous, but in other cases they're hard keratin, and those ones feel more like a grain of sand.
These are the ones been described as, and I know there is a difference between the cocoons and the shrimp-like or the worm-like.
There's all sorts of things, and then there's probably all sorts of things in between, and and some of them you just have to sort of sit down at the microscope and look at them and figure out what they are.
But altogether, there's only one symptom that is unique to Morgellons, and that is the microscopic fibers embedded in skin tissue, right?
Yeah, that's how we define it, even though there's other things, because that seems to be the one feature that is found in Morgellons that is not does not occur in other conditions.
Now, when you started researching Morgellons, you took a look at bovine digital dermatitis. Um, is what? How is that condition similar, or how did you think that condition was similar to Morgellons? And can you elaborate a little bit on the difference between the two?
Really, I got into researching Morgellons because of bovine digital dermatitis. I had
Okay.
heard about Morgellons, and I just thought that I thought, oh, that's weird, and I didn't give it much more thought of that. And I looked and I saw some videos, and there were some doctors saying, I'm sure it was one of the naysayers saying, yeah, see, blue fibers, I can pluck them out of clothing, and yeah, and they were making fun of it. And then I saw Dr. Stricker on a video, and he said, but look, there's fibers under unbroken skin, and no, these are not textile fibers. And I trusted Dr. Stricker's opinion because I knew him personally. And I said, well, if he thinks that this is not textile, then it's not. And, really, I didn't think too much more of it. And then I happened to go out for dinner with one of the vets I know, and he was telling me about a spirochetal infection in cattle, and the cattle had these unusual filamentous projections. And I said, and that it was associated with spirochetal infection. And I knew a lot of these Morgellons patients had borreliosis. So I thought, well, I wonder if some similar process is going on. So that's what, led me into that. And then I did propose to on a ILADS chat group. I told people about it, and I thought somebody else would investigate it, and he did. And I thought, well, in a nutshell.
Gotcha. So can you tell us, are the pathogens the same? Is it Borrelia that's involved in bovine digital dermatitis like Morgellons?
I have recently heard of a few cattle that actually have detectable Borrelia infection. However, in most of the cattle, they find that the bacteria that they commonly find are various species of treponema.
Oh.
Except for treponema pallidum, of course, because that's exclusively a human pathogen.
Oh.
But they find, different oral type spirochetes and, different spirochetes in the genus treponema. Those seem to be the key etiologic factors in bovine digital dermatitis. And, but at least seeing that gave me some clues of what I might look for in Morgellons tissue, because we knew that there was that, for one thing, it was known that the fibers were composed of keratin and that there was a defective process of keratinization occurring in the skin. And we also knew that the skin contained detectable spirochetes. So it gave me a starting point on what to look for.
Do the cattle get the fibers?
Their things tend to be a lot thicker and bigger. They're more spiky growths on the skin, but in some specimens, I do see little blue filaments that are pretty much identical to what I've seen in Morgellons. But, we haven't done enough experiments to really understand it totally.
More like harsh hairs. There were early reports of Morgellons fibers being discovered inside organs. Theoretically, in light of what we know about Morgellons, could such a phenomenon actually occur, where you're finding Morgellons fibers inside the liver, the kidneys, the heart?
I think anywhere there's epithelial tissue, you could probably find a similar thing. And, certainly all cells that have a nucleus, they encode all the different proteins in your body. So, if the right conditions were there, that, the genes encoding for those proteins were turned on, then, hypothetically you could have such a thing. I have not seen it in like liver or tissues like that, but I've known cases where women have had unusual keratin filaments that were vaginal, and men that have described them from their anus, or that seem to be associated with the rectum. And I also know a case of a Morgellons patient who had unusual ulcerations in her intestines, and then the intestines, the ulcerations and adhesions were fusing, causing a lot of problems. So it appeared that a similar process might be happening internally.
Wow. This bacteria really does a lot of damage, huh?
It does, and then it frequently does not strike alone. It's frequently associated with different co-infections and co-involved organisms. So, in Morgellons, I'm sure that even though the key etiologic agent is Borrelia,
Yeah.
in the cases we see, there are probably other co-involved organisms that make the condition more complicated and more difficult to treat.
Yeah. Well, before we get there, though, and I think you just said it, is Morgellons a condition or a disease?
Well, I would call it a condition because it's a symptom associated with that infectious process. So that's how I define it. But it depends on how you define a disease. So a disease is a disorder of structure or function in an organism, such as a human, that produces specific signs or symptoms and that affect specific locations and that are not simply a direct result of physical injury. So, under that definition, Morgellons is a disease. But, here's an example. If you look at measles, measles is a systemic infection. So that is a disease. Are the spots that a person with measles, are those a disease? So I would say they're more a manifestation or a symptom of the disease. And then the disease is measles, the viral infection itself.
Oh, gotcha.
So does that make sense?
Yeah, because measles is also a multi-systemic disease as well, right?
Exactly. So, the disease is measles. It is that viral infection. And are the actual little spots that they're really more a symptom of that disease, right? Well, that's how I think of Morgellons too.
So the disease is really Lyme disease or borreliosis.
Lyme disease and associated diseases, because you might have a different constellation of infections in different patients. And they may all be co-involved in the Morgellons symptoms. Does that make sense?
Oh, yeah. Yeah, absolutely. And we're going to talk about biofilms in just before we get there, though, I got to ask you, how does classification and staging work?
Well, yeah. Let's see.
It sounds like.
I'm sorry, I got a weepy eye, and it's driving me crazy. I get a tear duct, and it's so I'm not crying, everybody. I'm just this thing. It's so distracting and annoying. But anyway,
sorry, my friend.
Yeah. Sorry. I'm just gonna wipe my eye a second, if it's okay.
Take your time. I want to thank everybody who's on the chat right now. Lance, Clay, Shelly. Hey guys, how you doing? Everybody is definitely appreciative of you making time to be here today, Rhea. And if you have any questions, guys, feel free to drop them in the box here. We'll try to get to them, time permitting. And
here we go. I'm back. So really, the classification of diseases,
Sure.
it provides a foundation for identifying causation of morbidity and mortality. And it really provides a common language in which different healthcare professionals can communicate with each other. And, you know, theoretically, you want it to be able to assist in lessening suffering and, helping to improve the quality of life and saving lives. So, we did propose a classification scheme for Morgellons, and this was based on early lesions versus late lesions and localized lesions versus disseminated lesions. And staging uses it's different in that it uses objective medical criteria to assess the disease progression or um, you know to assess severity and disease progression in a disease. So our staging system for Morgellons, we separated it into mild, moderate, and severe cases. So together we hope that staging and classification will help validate Morgellons.
So this is really kind of like a manual for the disease that doctors can use to aid in their diagnosis and treatment. But
Correct, and then in identifying it too, they can use it to help identify and diagnose and treat the disease.
do do they have to do anything special to start using this classification and staging system?
For the classification, not really, because that's it's based on whether a lesion is early or late, or but to assess the severity of the disease, they need to do histology, and they need to look specifically for Borrelia infection to
Hmm.
see where the infected tissue is and the extent of the infection. So yes, the staging, our proposed staging does require special processing. And unfortunately, it's not readily available in most laboratories.
Yeah, so they would have to send specimens off to a special laboratory or order
And
the.
yeah, and to a laboratory that's capable of staining specifically for Borrelia infection.
Okay, and that is kind of a special process itself, right?
It is, you'd have to do immunostaining or fluorescent in situ hybridization or some other staining method. In our staging system, we used immunostaining.
Okay, okay. Hey, thank you for making time to elaborate on each of these questions. We did talk about immunostaining. Tell us about biofilms and these constellations of infections.
So biofilms are communities of bacteria that are enclosed in a protective matrix. So, in Morgellons patients, we've seen some Borrelia biofilms, and some of these are mixed infection with Helicobacter. In that particular study, we were looking for those two microorganisms, but we have also found in some patients rickettsial species and Bartonella henselae, and of course Treponema denticola. So, and tick-borne infection there's quite a few different pathogens that are associated with tick-borne infection. So hypothetically, there could be other microorganisms involved too. So, we need to research what different pathogens are involved in Morgellons better, and have a better understanding of exactly what kind of diversity we have in terms of bacteria in Morgellons tissue. And whether these are mixed infections or exactly what's happening in skin.
These communities help enable persistence.
They can, because they are enclosed in that protective matrix. So, antibiotic therapy is not as a is not often does not it's not able to penetrate that. Also, intracellular infection can be very difficult to treat too, because not all antibiotics can, are not all of them are effective intracellularly. So, yeah, the organisms have a lot of different tricky ways to, protect themselves, and um, so that they can persist.
Yeah, including genetic variants on their outer surface proteins as well.
Yep, they've got lots of different mechanisms.
Tricks, bag of tricks.
Hmm.
Um, the CDC, and this was a win, recently updated their website to warn about congenital Lyme transmission,
Hmm.
but not regarding sexual transmission.
Hmm.
Can Lyme infect patients in the same manner as syphilis?
We believe so. Um, we did research showing that we could find Borrelia spirochetes in vaginal and seminal secretions. And, we're not allowed to go around experimentally infecting humans, but we were able to culture live organisms from, those fluids. And we do know that there have been animal studies in which sexual transmission was, definitively shown. One of these was a study in dogs done at the University of Wisconsin. So the dogs, experimentally infected male dogs, were able to transmit Borrelia infection to uninfected female dogs. And then those female dogs transmitted the infection congenitally to their pups. So, I think that because of the animal models, there's a lot of evidence to suggest that this too may happen in humans.
And you also did a little bit of research yourself, which demonstrates these infections in the sexual fluids. Is this correct?
Yes, we did. In
Okay.
vaginal and seminal secretions, we could find the Borrelia spirochetes in those fluids, and then we also successfully cultured them. So when you culture it, there's active cell division happening, so you know that that organism is alive.
That's a current infection, not a past infection.
Exactly. If it's able
And
to grow and reproduce, then it is alive and it is present.
and just to be clear, the patients that you studied in that, or the subjects that you studied in that research, were married couples. And
Some of them were, yeah. There were mm, there was a subgroup that were married couples, and in those they seemed to have identical strains of Borrelia.
right,
So three of them, I believe, had Borrelia burgdorferi, and then one couple had Borrelia hermsii.
gotcha. Okay, two different people, but identical strains of supposedly
Yeah,
rare
yeah.
bacteria. Okay, thank you for elaborating on that. Um, PCR analysis, does it simply amplify which agents you're looking for, or does it actually give you a unique answer?
Well, when you do PCR, you have to use a primer that is specific for your target. And, different, some proteins are found, in some sequences are found in several different organisms. So it might not be that specific. So related organisms like maybe treponema and Borrelia, if you look at something like the flagellar protein, that might be highly conserved between the two groups. That may be less specific than an outer surface protein. So again, it's like immunostaining. You have to look at the sensitivity and the specificity of your primer.
Okay, so you do actually get an actual answer. It's not just whatever you're looking for.
No, and it, it depends. You have to, and sometimes some strains of Borrelia are very specific for one target, and so
Oh,
sometimes we have to use several different primers in order to find Borrelia. So, you can't use just one primer and then if it's too specific, for example, for Borrelia burgdorferi or even certain strains of Borrelia burgdorferi, it may totally miss some infections. So you really have to um, look at the specificity of your primer and understand it.
gotcha. Well, if you could direct a Morgellons study with unlimited funding, what would you intend to discover?
Well, possibly more about the mechanism of what is happening, and how what's happening at the with gene regulation within the cells and that sort of thing. But also we want to find out more about the different pathogens that are associated with it, and we'd also like to understand better the secondary etiologic factors. It seems to be, is it linked more to a specific group of people? Is there a genetic factor? Is there genetic predisposition to develop it? And um, are there certain strains of Borrelia that are more likely to be linked with it? There's so many, things that we would like to find out. But basically, there needs to be a lot more done. But unfortunately, with it being so controversial, there's not much interest in people to study it. There's
Hmm.
very few of us that have the interest.
Yeah, that's a frightening scenario that the research could just suddenly one day stop.
And I mean, speaking for myself, I get overwhelmed. Sometimes I have too much on my plate. I do it all as a volunteer, so it's sometimes I just can't do everything I'd like to do.
Mm.
I just physically I don't have the time, and I do as much as I can, but I've spent a few years where I was working on it almost like a full-time job.
Wow.
I don't, I'm not paid for it. It's purely voluntary. So,
Yeah, and your work is invaluable. The facts that we now have about the condition, we wouldn't be anywhere today without it, without your efforts.
well, without a lot of other talented people who have collaborated with me, I'm one person. I couldn't possibly have learned all I know on my own. I depended heavily on collaboration. So, there's a lot of very talented people that have been involved with it. There's people at University of New Haven, of course, Dr. Stricker, Dr. Karsten Nicholas, but there's other collaborators too that are too numerous to count, and also a new collaborator, Dr. Vet Lloyd at Mount Allison University, and
Oh,
we've been fortunate to have so many talented and wonderful people who have dedicated their time and their skills to this as well.
yeah, we're definitely blessed to have so many talented people working towards this.
Yeah, I don't like to take credit for for my contributions, because my contributions are part of a larger team effort.
Well, humility is definitely an admirable quality, my friend.
Well, thank you.
Um, so what's you currently working on?
Um, with Morgellons at present, I have an interesting case study on the go, but with the COVID situation, I haven't been able to work with the pathologist on it because we have to share a microscope. So,
Hmm.
we're postponing that a bit. And then some of the universities that I collaborate with have also shut down, or are not in full gear. So, that slowed things down a little bit. But, we are working on identifying some of the, with Dr. Eva Shapi's research group at the University of New Haven. I've been working with them on, finding some of the different pathogens that might be present in Morgellons skin. And I've been helping Mount Allison University a little bit with things associated with the mechanism.
So coronavirus really directly impacted your ability to research.
Yeah, a little bit. I mean, I could still send samples, but the collaborative effort just can't, with universities and things shutting down, definitely affects things. And
Man.
yeah, and I mean the sharing of knowledge and findings, that's been affected too with for example, the cancellation of more the Charles E. Holman Foundation. Um,
Annual conference.
yeah,
Yeah.
conference. That was a real shame. But I'm so glad that you gave us the opportunity, to share some findings with the larger community.
Oh, thank you for giving me the opportunity to interview you about this.
You're welcome.
We've got
And
a lot.
sorry about the crying. It's just,
This was a very emotional episode. No, I'm just kidding. We know you got the tear ducts.
it's annoying.
Well, we're just about done, my friend. I think we got a couple more questions and then we can let you go. And thank you so much for making time here today.
You're welcome. My pleasure.
What's the most common question people ask you about Morgellons?
Most people that contact me are pretty well informed, so they tend not to ask me about the research things themselves, but most of them are patients and they want to know, where can I go to get treated? Where can I go to get diagnosed? Which is really difficult because it is controversial. It's not widely accepted. So, my answer to that is the Charles E. Holman Foundation can sometimes give some information. I know a little bit about, where help is available in Canada. Um, but, really, because it's associated with Lyme and Lyme itself is controversial, your options for treatment and diagnosis are limited wherever you go. So,
All right. And so the best thing people can do to help you is to donate to the Charles E. Holman Foundation, and then we can get some more research accomplished on the condition.
definitely. And I mean, not all of it goes to my research, but if other people come up with some good research ideas, I know that the Charles E. Holman Foundation is very open to, other research groups too, should they happen to come up with something that looks promising and interesting.
Commendable. Marianne, thank you so much for being here today.
Well, thank you so much.
And we hope to have you back on the show. If you guys like this interview, feel free to share it out with your friends. And we definitely look forward to seeing everybody again soon. Marianne, take care, my friend.
Thank you. You too.